Hey everybody, we are looking at today, finally, going back to section 12.1, what we normally, cover at the start of a chapter and not kind of in the middle. So last time we looked at section 12.4, which was all about meiosis and that kind of process of events. Here with 12.1, we're going to go into kind of the first real part of genetics and inheritance and looking at exactly how traits get passed down from one parent, or from two parents, I should say, to their offspring. And the scientist that is most responsible for understanding of that is a man by the name of Gregor Mendel, who happens to be one of my all-time favorite scientists. Like my Google image, if you've ever wondered who that is, like that little black and white picture on my Google account, that is Gregor Mendel. He is the father of genetics. And so I like talking about, as I've told you before, genetics in general, but Gregor Mendel is super interesting, I think. So this is where we are. We're going to look at kind of the history behind this here in section 12.1, and then move into things like Punnett squares in the coming days. All right, so go ahead and get the title if you have not already. And in terms of our objectives for this section, we just have two. So looking at where an organism, how it gets its unique characteristics, the likelihood, the probability of that happening, and then looking at how different forms of a gene get distributed to offspring. So kind of the separation of those, what we call alleles, in the formation of things like gametes. So here's a picture of Gregor Mendel, and this is very similar to the one that I've got on my Google account. But in looking at Gregor Mendel, a couple things that are kind of, I think, interesting about him. He was alive back in the 1800s. We're going to talk about Charles Darwin a little bit later on in the year, and Gregor Mendel did his work around the same period of time. Gregor Mendel, unlike Charles Darwin, though, was not a scientist by training. He went to school like normal, but at that time, if you were of a poor community, you did not have access to things like going to college. You had to be wealthier to be given kind of that opportunity. And so Gregor was raised, I think, by farmers in Austria at the time. But he was very, very interested in the natural world. And he was very interested in things like farming, like how do you improve upon yields of crops and those types of things. Gregor took a position as a monk in a monastery in what's now the Czech Republic. And in that role, his job was to maintain the monastery gardens. And so as part of that, he spent a lot of time with plants and other organisms in the garden. And he became very interested in trying to figure out exactly how and what caused certain plants to have certain traits. And the plants that he focused most on for his work, specifically aligning with what we're going to be talking about in genetics, were pea plants. All right. So he's called the father of genetics because he was really the first one to actually kind of concretely study and come up with any type of valid conclusions about how things are inherited, how things get passed from parents to offspring. So genetics is just that, study of inheritance patterns and variation in living things. When we talk about genetics, we use the word traits a lot, and these are those characteristics that we obtain through our chromosomes from our parents. All right, so if we think back to the 1800s, keep in mind, people didn't have any real idea about how things got passed down. They knew that parents and offspring tend to kind of look alike, but they didn't really understand why. And they had pretty strong assumptions that a lot of what we see is just simply blended. But we know, based on the fact that, we have scenarios where, for example, two brown-eyed parents can produce a blue-eyed child, that it's not always as simple as just blending. All right, and so what he's essentially going to teach us is what we now have learned and to call like dominant and recessive, and we'll get to that in the next section. All right. But Gregor Mendel laid the foundation for our current understanding of genetics. He showed that traits are inherited as discrete units, which are what we now refer to as genes. So it's not that traits are just simply blended, but there is a sequence, there is a code. It is like a recipe to make these specific traits, what we see on the surface, okay? So these discrete units become super important because they help us to kind of start thinking about how this process actually works. So in terms of his studies, I mentioned pea plants were his kind of number one choice. They were easy for him to work with and manipulate. And with all things plants like animals, they're generally sexual reproducers, which means they have male and female gametes. And so as part of this process, he had to make some pretty important decisions. And these are three that we tend to attribute to his success, meaning if he hadn't done these three things that you're seeing here as listed 1, 2, 3, chances are we wouldn't be talking about him today. Because like I said, he wasn't a trained scientist. Nobody really knew who he was. And because they didn't really know who he was, they didn't really trust him at the time. It wasn't until, a century later that we finally started to understand, oh, this guy actually did some impressive stuff. So his first really important decision was what you see there for number 1. He used true breeding or purebred plants. And what that means is that these are plants that have consistent ancestors, right? If you are going to get a dog and you wanted to have a purebred German Shepherd, right? We know the background, the historical background of that family line. That's the idea. This is part of the reason why it's more expensive generally to get purebred animals and purebred plants if you're a farmer, because they have to ensure that there's been no cross-contamination, okay? So he used these purebred pea plants. He knew that there was nothing that was kind of crossed between the species of pea plant he was using and other species of pea plants. He also, in his second important decision, he controlled breeding, which means he had to kind of modify the fertilization process. And we'll talk about that on the next slide. But as I said, this is sexual reproduction. And so with a plant, that involves some physical manipulation of some things, okay? He did create what we call hybrids, and that's a vocab term here. Hybrids are things that express traits from both parents, right? So he crossed 2 true breeding or what we would refer to as homozygous types of plants with 2 different traits. And he was trying to see the mixture between those and how that would kind of pan out in the offspring. He also chose in pea plants to observe 7 either-or traits, and we'll talk about exactly what those were in the next couple of slides, but they included things like pod shape, plant height, pea color, flower color, and so on, okay? The benefit in choosing those either-or traits was that really he didn't run the risk of maybe missing something, right? These are traits that are only dominant or recessive. We know that in the case of most living things, things are not just simply either or. Like in humans, for example, you don't just have brown eyes or blue eyes. There's lots of variation in between. You don't just have red hair or blonde hair. There's other variation and other options there. But the traits he chose to work with just happen to be those traits for which there's no kind of intermediate phenotype. It is one thing or it is the other, and there's nothing in between. So the way he was able to control breeding, number 2, right here from this last slide, what we're seeing here on this slide, was he actually used like little tiny scissors and paintbrushes. Sounds silly, but I mean, we're talking about the 1800s, right? So he didn't have access to any real scientific research and lab equipment. So what he used as part of this process, what he would essentially do is he would take the male parts, which are called the stamen. These are these taller parts of a flower that kind of stick up sometimes. And if you've ever kind of bumped into them and they're sitting maybe in a vase on a table or they're outside even, you may notice that the pollen that's sitting on those, pollen, by the way, is the male gamete of a plant. It's like sperm for animals. But that pollen just basically brushes off, falls off, and the idea is that in the plant, within this flower, that pollen will fall and fertilize the egg, which is stored here in what we call the pistil, or the female organ of the plant. All right, so he would remove the stamens, being careful not to get pollen everywhere, and then he would take pollen from whichever plant he was interested in looking at, and take that pollen and brush it onto the pistil, the female reproductive organ right here, of that same plant. So he was preventing these plants from doing what plants do on a regular basis, which is called self-fertilization, where the pollen essentially, like I said, falls down, fertilizes, and that whole flower is then going to reproduce and make seeds, right? He didn't want that to happen because he wanted to be able to control and actually see exactly what he was interested in seeing. Okay, so he would choose specific plants with specific traits and cross them. So he removed those parts and he would paint the other ones on. And in so doing, he developed different names for these generations of plants that he was working with and creating. So the first generation he started with was called the parental or P generation, not for pea plant, but P for parent. And that first generation was used to create a first generation of offspring. Okay, my first generation of offspring are what he's going to call the F1s, or the first filial generation. Those F1s, in his experiments, he allowed to self-pollinate, to self-fertilize. So he didn't do the paintbrush thing for that second generation. He just allowed the F1s to self-pollinate, and then the offspring that resulted from that were what he called the second generation of offspring, or the F2, second filial generation. Okay. F1, F2, and P are going to be terms that you hear often, especially when we get into looking at Punnett squares and doing some actual crosses. So here's one example of one type of experiment he would have done, which is to look at flower color specifically. All right, and in looking at flower color, pea plants can either have purple flowers or white flowers, and that's it. If he crossed a pure breeding purple flower only pea plant with a pure breeding white flower only pea plant, what he noticed was that all of the offspring that would result from that were always 100% of the time purple. Okay, and he found that kind of odd because it seems as if the white trait just kind of disappeared, right? Well, if he allowed these F1s then to self-fertilize or cross two F1s together, what he always got for his results was also kind of interesting. It was like the white one all of a sudden reappeared in one of the four offspring. So 25% of the time, and it's not that they have four offspring every single time, but 25% of his results of the offspring produced in the F2s ended up having that white trait. So to him, it was very odd. It was like the white is present here. Somehow it disappears here, but then it reappears in this third and final offspring generation. It was as if it was being hidden, okay, as part of this process. So these were the different traits that he looked at. These were the types of what we call crosses that he did. We have pea shape, pea color, flower color, pod shape, pod color, flower position, and plant height. Okay, and in general, the ones, this isn't necessarily true. I was going to say the ones across the top are the dominant, but that doesn't really fit with this either. But those are his 7 either-or traits, and in every scenario where he did these types of crosses, he always ended up seeing, again, 100% of the time, one phenotype being shown. Okay, one what we now call dominant phenotype appearing in the offspring. So what we see as a result of this, and I guess I do have a text box here, so let me pull that up, is, again, in this case, here are my parents. Those are my crosses. And again, in his F1s, he's seeing only one trait appearing. One trait is actually being shown. He's not getting both yellow and green peas. He's only getting the yellow ones, and so on. So for those, we're only seeing that one parent's traits appearing in that F1 generation. But when he does it again, like when he crosses those F1s again, like we see right here, let me scoot back, he's going to see this yellow, in this case, trait appear again in the F2 generation. So when he's considering what's happening down here where I'm getting these F2s, again, he's getting this ratio of 1 of this kind of uniquely hidden trait to 3 of these more commonly seen, more frequent, what we're going to call dominant trait. Okay, and that was consistent regardless of which trait he was looking at. If it was flower color, if it was height, if it was flower position, if it was pea shape, it did not matter. They all kind of gave him the same general mathematical ratios. So he drew some important conclusions from what he did here. The first one was that traits appear to be inherited discretely or as individual units, what we now call genes. That means we're not blending things here, right? There's an equal probability for a pea plant that has all these traits with, maybe purple flowers and yellow peas and wrinkled pods and so on. Each of those is inherited independently of one another. Okay. We know that organisms inherit 2 copies of each gene. One comes from each parent, and those are those homologous pairs that we talked about with section 12.4, the homologous pairs that we separate as part of anaphase 1 in meiosis 1. Okay. So those 2 copies of each gene come from each parent. Those copies are passed down in gametes, and when those 2 combine, we end up with diploid somatic cells that have 2 sets or 2 N chromosomes. The third and final conclusion that he drew from this is that our two copies of these homologous pairs are going to segregate or separate during gamete formation. This is called the law of segregation. And this simply means that those pairings that you get actually do separate when you go to create your own gametes. What you got from mom and dad are going to be pulled apart and separated and end up in two different gametes as a result of meiosis, and specifically meiosis 1. Okay. So that separation process is something that he had to kind of come up with here in seeing that, this trait is somehow present and then it's gone and then it reappears down here. So somehow it must be kind of hidden here. Someone must have passed it down and it must have been present in this generation. It's just it wasn't shown until the next one. All right. So that's all I've got for 12.1. We'll come back and talk about Punnett squares next time. But as always, if you need anything, let me know. Otherwise, I will see you later. Bye, y'all.